So in this video I will start with the smear preparation and I will show you the simple staining, direct staining. First you incinerate your loop inside the incinerator until you see that your loop is orange in color, hot orange in color. I'm going to be using bacteria from solid culture. Therefore I need a drop of water to be transferred to my loop before I start working. To my slide before I start working. I will dry my slide on filter paper and I will transfer a drop of water to the slide where I'm planning to work. This is enough water. This is the amount of water necessary for you to prepare a smear. I re-sterilize the loop. So until I see the loop itself orange in color, then I leave it for a few seconds to cool down outside the incinerator. My Petri dish, I always put it on the bench, lid down. This is the media, those lines are the bacteria, and I put it lid down. When I open it like this. I touch the places where there are no bacteria to make sure my loop is not too hot. I take a little bit of bacteria and I close it back. So this is why we put the lid down. So I open it near the heat, take bacteria and close it back. This bacteria that I have taken, I mix it with my drop of water and I spread. This is the first step of smear preparation, the spreading. As we have discussed, or as I have mentioned in the pre-recorded lecture, second step is air drying and then heat fixation. I spread as much as I can. The further the bacteria from each other, the better it is. I re-sterilize the loop and I keep it aside. And this smear has to be left now for air drying. I leave it like this for air drying. My smear is dry now. If you look at it, it's milky in color. It's very important to label your slide with a permanent marker when you start working on it, so you wouldn't be confused later on where is the smear on this side or on the other side. Now I need heat fixation. I will use the incinerator for heat fixation. I touch the slide and I count to 5. 1, 2, 3, 4, 5. So I'm holding the slide from one end and I am touching the incinerator. I do this on both sides 3 or 4 times. Heat fixation kills my bacteria, coagulate the protein of the outer membrane so the bacteria get fixed on the slide, and it helps to deactivate the inner enzymes to avoid autolysis. Now I am ready for staining. In this video I will show you the direct staining. I have chosen for you methylene blue. You can choose any alkaline stain available in the lab. I will cover the smear, all of it, with the stain, and I will count to 30 seconds. After 30 seconds, I will need to wash off the excess stain from the slide. So I incline the slide in the wash bottle and I wash the excess stain with distilled water. Now my slide is ready to be focused. I tap it on filter paper to dry it and I start focusing it under the microscope. I put it on the stage, fix it with the stage clips, move my stage in a way that I put in the spot of light a good part of my slide. A good part is not a very dark part and also not very light. With the coarse adjustment knob, I take my stage all the way up and I have clicked in place the lowest magnification, which is the 4x in our case. I look at the light if it's good or no. If it's good, I start focusing. I'm lowering the stage slowly while looking to my slide until the field in front of me is clear. Once it's clear, I click the next magnification. I stopped using the coarse adjustment. With the fine adjustment, I clarify the image. Click next magnification, fine adjustment, make the image clear. Now I need to move to the oil immersion. Can I have oil please? I need to move to the oil immersion. So I increase the light to the maximum and on the spot of light I put a drop of immersion oil. Then I click and place the highest magnification, fine adjustment, I clarify the image, and this bacteria is bacillus. They are bacilli in chain. I'm gonna show you pictures of them on Blackboard inshallah.
Stop.
Again, I don't want to crop the videos. Stop.